Athletic success isn't just about your physical performance, it's also about the beneficial effects that exercise has on your body, both physically and psychologically. Have you ever felt the overwhelming sense of joy after exercising and wondered why it happens? Historically, we thought the runner's high was all about endorphins. However, recent evidence is slowly revealing the real champion behind runner's high, the endocannabinoids, and how this phenomenon is heavily influenced by genetics. Today, we will dive into the science behind this fascinating intersection of genetics and sports performance. Welcome to the genetic basis of stuff and things. So, what exactly is a runner's high? In 1979, Professor Gary Egger investigated the relationship between exercise and pain. After running around the cricket ground, he demonstrated increased pain tolerance to the inflating blood pressure curve. These fascinating pain-relieving effects were noted as evidence of ecstatic states and anesthesia associated with running. Later, these effects, along with the feeling of happiness, relaxation, the reduction of anxiety, and relief from pain after exercising, became popularly known as runner's high. In the past, the spotlight was fully on endorphins, a neuropeptide which is responsible for the perception of pain and feelings of well-being. There are more than 20 types of endorphins in your body, with mainly beta endorphins being involved in stress relief and pain management. Endorphins bind to the opioid receptors in the brain, which are present on the surface of neuron cells to receive the signal, helping to slow down messages between the brain and the rest of the body. Therefore, it was believed that the pain relief during runner's high is related to the delay of signals during message transmission. Despite numerous studies suggesting increased endorphins in the blood during exercise, these studies did not directly prove that the endorphins are affecting the brain or the central nervous system. It was also found that endorphins aren't even able to pass through the blood-brain barrier to reach the brain, unlike a related range of molecules called anandamides. Anandamides belong to a class of molecules called endocannabinoids, and they bind to the cannabinoid receptors such as CB1. CB1, which is encoded by the CNR1 gene in humans, is responsible for receiving signals on nerve cells in a wide variety of regions of the brain. When anandamide binds, it modulates the release of neurotransmitters and regulates communication between neurons. Since anandamides dissolve easily, it can travel to the brain where it binds to the CB1 receptors, which is believed to reduce anxiety. Although the endocannabinoid system is highly conserved across mammalian species, genetic variations within the system can affect how pronounced the runner's high is. For instance, some common genetic variants in the CNR1 gene is associated with increased sensitivity to major depressive disorder and antidepressant treatment resistance. In the endocannabinoid system, an enzyme called fatty acid amide hydrolase breaks down endocannabinoids. A common variation in the fatty acid amide hydrolase gene results in an amino acid change from proline to threonine at residue 129. The threonine variant leads to a reduction in fatty acid amide hydrolase activity, allowing more anandamide to be present. Individuals carrying this allele therefore exhibit reduced reactivity to threats and quicker habituation to repeated stresses, possibly due to enhanced endocannabinoid signaling. This suggests that individuals with the threonine allele may experience a more pronounced runner's high. So, how does this all work? In 2015, an experiment was conducted where 32 mice were provided with running wheels to investigate the role of endocannabinoids in runner's high. The mice were assigned to two groups, a control group and one subjected to wheel running. After they were tested for anxiety-like behavior in the dark light box test, runners spent increased time in the aversive bright area compared to non-runners, indicating little signs of anxiety. The runners were also less active and displayed fewer exits from the dark compartment into the lit compartment. Then, these same mice were subjected to a hot plate test, which was used to study pain sensitivity. The runners are less likely to lick their hind paws or jump as their first action when put on a hot plate, suggesting their reduced thermal pain sensitivity. Hence, this study shows that acute long-distance running reduces anxiety and pain. 2 days after behavioral testing, endocannabinoid levels were measured across the entire body. It was found that running significantly elevated endocannabinoid levels in blood plasma. However, levels of endocannabinoids in the brain were only marginally increased. The researchers hypothesized that the lipophilic structure of endocannabinoids may have allowed for their rapid distribution and elimination throughout the body, resulting in lower concentrations remaining in the brain. This idea was supported by a separate study which found that long-term wheel running in rodents led to significantly increased levels of anandamides in the rodent hippocampus, a complex brain structure embedded deep into the temporal lobe. So, the runner's high isn't just about endorphins. It's a complex interplay between neurotransmitters and endocannabinoids, influenced by genetic factors such as endocannabinoid receptor gene CNR1 and the fatty acid amide hydrolase gene polymorphism. From the thrill of a runner's high to the raw power of a sprinter, genetics influence every aspect of athletic success. The next time we feel that successful euphoric rush and pain relief after exercising, remember, it's not just one thing but a combination of body-generated chemicals and genetic variations working hand in hand with each other.